Hello, and welcome to the Comparative and Omics Tools in BioCyc webinar series. In this series, we'll be taking a look at what you can do with single organisms or across multiple organisms to look at omics datasets or to do comparative genomics type studies. In part 1 of this series, we'll be looking at the cellular overview, the metabolic snapshot of a given organism. I'm Alexander Shearer, speaking on behalf of the Bioinformatics Research Group at SRI International and for BioCyc.org. If you haven't yet, I encourage you to go take a look at our Introduction to BioCyc webinar series, because that's going to explain a lot of the basics of the pages that you're going to be accessing and seeing as you go through this webinar series. In the following parts of this series, we'll be looking at additional omics and comparative tools. In part 2, we'll be looking at how to use the cellular omics viewer to display omics data. And later on in part 3, we'll be looking at comparative genomics in BioCyc. And in part 4, we'll be looking at ortholog viewing across multiple organisms in BioCyc. And in a future webinar, we'll also be looking at some of the exciting new tools that are available through the desktop version of Pathway Tools and will be available through the website later. These include things like displaying omics data on the genome and a whole new way to look at the genetic regulatory network. In this first part of the webinar series, we're just going to be looking at the cellular overview diagram. And as always, I like to start on the BioCyc query page because I can access whatever I want from there. And you can see, and this is actually new since the previous introductory webinar, that we now have little icons to tell you where you can click on to get the genome browser or the cellular overview diagram. So I'm just going to click on that over here right now, diagram. And that takes us to the cellular overview diagram, in this case for E. coli, because that's our default organism. Okay, so a little background on the cellular overview diagram. This is a means to quickly visualize the metabolic complement of an organism. It's generated at the touch of a button by the Pathway Tool software, and what that means is that if you're making your own database or anytime we update a database, we can actually just tell it generate a new metabolic snapshot for us based on the changes we've made. So this isn't anything we've painted or drawn or done by hand. This is generated dynamically at our request by our software. And the upshot of that is that you can get one of these for any organism that you make a database for without having to go through a lot of muckery to draw it yourself. The cellular overview diagram can be used both as a static display of the metabolic network and to visualize large-scale datasets. That second thing is what we'll be talking about in part 2 of this series. For now, we're looking at this as the static snapshot of the metabolic network. So let's talk a little bit about how the cellular overview diagram is organized. Okay. So we can see that we clicked through from the query page, the diagram opened. If you had an eagle eye there, you may have noticed that it said something about taking 2 to 4 minutes to load. That's us doing a really highball estimate. Actually, it usually takes seconds to load. So the cellular overview diagram is just a graph depicting the organism's biochemical network. It's that familiar kind of lines and nodes thing that you'll be used to seeing from wall charts and from reviews and from textbooks. At the top of the page, we have 3 links. These links go to 1, a set of instructions on how to use the page in case you ever forget anything or need a little more detail. 2, the Pathway Tools query page, so back to where we started from. And 3, the omics viewer that lets you paint omics data onto this diagram. And again, we'll be talking about that in part 2. And just to clarify, by omics data, we mean any large-scale data set that assigns numbers to genes, reactions, proteins, or metabolites, or things like that. As you'd expect, these lines, or edges if you're feeling more computational, represent reactions, and that includes both metabolic and transport reactions. So for example, if I move over a metabolic reaction here, and you'll see a little thing popped up there that we call a tooltip. I'll explain those a little later, but these are metabolic reactions. We also have transport reactions here that show how something, in this case cysteine, can be transported into or out of the cell. Most of our pathways flow downward visually on the page, meaning that you're looking at an input up here, in this case L-aspartate, and an output down here, in this case NAD+. We have a legend over on the right, and so I will just mouse over here. On the right here is our legend that explains what you're looking at. So triangles, amino acids, squares, carbohydrates, diamonds, proteins, etc. And things that are filled are phosphorylated forms. And as you noticed, you can mouse over stuff and get additional information. So right now I'm going to page down and across a little bit, and this thing here ought to be really familiar to you. So I've moused over a compound in the TCA cycle, and by mousing over it, a tooltip, this box, comes up. It tells me the compound I'm over, alpha-ketoglutarate, the pathway it's a part of, the TCA cycle, the super pathway in this case, which is the super pathway of glycolysis, pyruvate dehydrogenase, etc., etc. Super pathways are larger groupings of pathways into, as it says, super pathways, larger pathways that sort of carry out an overall process. So if we go to the top here, we'll see that we're on the pathway of glycolysis, which is part of this super pathway that you're familiar with from your introductory textbooks. And in addition to those basic tooltip mouse overs, if I again go down here to, let's go to isocitrate, then if I click on this, it brings up this expanded version of the window. And let me just scroll down a little bit so you can see it. And here we have, again, the information of what pathway we're in, the super pathway, the compound, and this big expanded view of the entire super pathway that includes the TCA cycle with all the genes listed, with the intermediates listed, and so on. And so if you need a little bit more detail without clicking through to the pathway, you can get it that way. I can also click through to the pathway. So for example, I can go up to this link here. Let's go to the compound, isocitrate, click on that, and it will actually take me through to the compound page for isocitrate. And this is just the straight vanilla compound page, just like we talked about in the introductory webinar series. And now I'll click back. And I'm back on this page with the overview, with this window still open. And to close the window, all you need to do is go over the close like that. So one more time, picking a different one, click here, we get the pathway of N-acetylglucosamine degradation, and I go over it here, close, and it goes away. So as easy as that. And if you're curious, the desktop version does something very similar with right-clicking, where you can bring up the exact same kind of thing. The desktop version actually has the advantage that you can generate additional windows at will and sort of follow them through from a pathway while keeping the overview open on your original window. Pathways in our metabolic overview are grouped very generally by function. Broadly speaking, we have anabolic pathways on the left, catabolic pathways on the right, energy pathways in the middle, and reactions that aren't in pathways on the far right. So if I click up here, you'll see this set of reactions here outside the cell, this set of reactions inside the cell. These are reactions that we've defined, in this case in BioCyc, that don't currently belong to a pathway. And there's actually a finer level of detail in how things are functionally grouped, but you'll kind of need to explore over time. But the upshot is that you should be able to find sets of related pathways living near each other in the pathway overview.